As a case study for understanding how membranes are so important to cells, we're going to consider this apparatus, which is a IV fluid bag that might be given to a patient at a hospital. So IV stands for intravenous. So that is to say a needle goes directly into a blood vein and it delivers liquid from this bag. So questions to ask yourself include, what is in this bag of fluid? And why is the composition of this fluid so important? As we explore the next slides coming up in this lecture, we're going to get down to how this answer may be solved. Before we get started, let's start here by describing gradients. And as I noted earlier, this is not the first part of Chapter 4. This material in your textbook is in the middle part of Chapter 4. So a gradient describes a difference between neighboring regions. And as a case in point, we can see we've got this black color fading to gray and eventually fading to no color, where it appears white. At the top, the black pixels are abundant and close together. We could say this is a high concentration of these black pixels. At the bottom, the black pixels are very sparse. It's a relatively low concentration of black pixels, eventually fading to no or zero black pixels present. We can describe this image as a concentration gradient, with a high concentration of black pixels at the top and a relatively low concentration of black pixels at the bottom. This arrow points down our concentration gradient. And we're going to often be examining diagrams where we see arrows pointing down a concentration gradient. And it's important to understand what that means. A cell's interior is chemically different from its exterior. Remember that cells are basically just little sacs of enzymes and water. And by using those enzymes, which are types of proteins, they can do tons of different types of metabolic activities. Cells can maintain this difference in its inside versus its outside by regulating the transport of dissolved substances called solutes across its membrane. And here we can see the arrows pointing across the membrane, and the solutes are moving from high concentration to low concentration. Solutes may enter or exit a cell by several different methods, and these methods really depend on two main factors. Number one, what is the concentration gradient? And number two, what is the chemical nature of the substance in question? That is to say, what is the polarity? What is the charge? What is the size of these solutes that are trying to pass through our membrane? We could really say that membranes selectively let things move in or out of that cell. And remember these words like hydrophilic and hydrophobic. Hydrophilic means water-loving, but hydrophobic means water-fearing. So this is going to be that region that's oily, that doesn't mix with water. This middle part is fatty, with nonpolar bonds forming, and the outside and inside of the cell has water, so polar bonds may form. To help us understand this, let's think about the club. So if we go outside the club or inside the club, the wall with the door is our cell membrane. So outside the club, the ladies line up, and the ladies want to get in the club. And of course, the bouncer lets all the ladies in. They go through the door, and they disperse throughout the club. And then the dudes line up, and of course there's a lot of dudes that want to get in, but the bouncer can't let them all in because there'd be way too many dudes. So he selectively lets the dudes in so that we have an even ratio of men to women. Now, what is the selective nature of this club door? Well, in this case, the example is all about gender balance, but in cells, it's all about the size, the polarity or the charge of those solutes in question. These membranes are composed of phospholipid bilayers, and as I've said, they selectively let things in and out. But remember those big proteins, those big purple blobs that are often represented in our diagrams? Those can also play a role in allowing substances to travel through and across these membranes. And as we saw previously, these proteins have a tremendous diversity of functions. This diagram shows us just some of those functions that are very useful for living cells. So to help us further explore this idea that solutes travel across membranes, we need to learn these 5 basic concepts for how this is possible. We have to learn about passive transport, osmosis, active transport, exocytosis, and finally endocytosis. To begin, we need to understand diffusion. Diffusion is a type of passive transport. It's passive because it requires absolutely no energy in order for this transport to occur. We can think of diffusion as a spreading out of particles or a spreading out of solutes that are dissolved into water. As a case in point, if I have a beaker of water and I drop in a little tablet with color in it, that tablet will dissolve and that color or dye will diffuse throughout the water. Initially, that color will be concentrated just around the tablet, but over time, the color will diffuse throughout the water and the water will appear as purple. Passive transport involves the movement of molecules from high to low concentration. No extra energy is required, and it may or may not use those protein channels, those big proteins embedded in membranes. The direction of this movement is from high concentration to low, which is to say down a gradient. Think about a cyclist. They start up at the top of a hill and then they slowly coast down that hill. It doesn't take any energy to go down the hill because the direction of the movement is from high to low. The next way in which substances might move across membranes is osmosis. But osmosis is very specific because it's only about the movement of water molecules. Osmosis is the passive transport of water across the membrane. And when we're talking about osmosis, we have to use these words that you see on your screen: hypertonic, isotonic, and hypotonic. When we describe these cells, we need to talk about the environment in which that cell is found. When we place a cell into a hypertonic environment, the water is going to move out of that cell. When we place a cell into a hypotonic environment, the bulk of the water is going to move into that cell. And when we place a cell in an isotonic environment, water moves into and out of the cell with no net gain or no net loss of water. So make sure you're looking at your screen right now. Here we can see these 3 cells, and notice the word water. It's either really big or it's really little. And now I'm going to click to the next slide. There we see. When we place a cell into a hypertonic environment, water moves out. When we place a cell into a hypotonic environment, water moves in. And when we place a cell into an isotonic environment, the water's moving both directions at relatively the same rate, so we don't have a net gain or a net loss. Let's talk about this now in the context of ourselves with our red blood cells. Our little red blood cells are these red discs. They kind of look like little donut shapes. And when a red blood cell appears normal, it looks like the picture on your screen. It's a nice little flattened disc. And when we put red blood cells into an isotonic solution, they're very happy. What if we place our red blood cells into a hypertonic solution? What's going to happen? Well, there we go. Those red blood cells shrivel up because water leaves the cell. And that's a bad thing. You're not going to be healthy and happy if your red blood cells are shriveled. What if we place our red blood cells into a hypotonic solution? What's going to happen? There we go. Those red blood cells swell, and eventually, if they are placed in a hypotonic solution for too long, they can even burst. Because remember, red blood cells are in our bodies. They're animal cells, so they have no cell wall. There's nothing preventing that cell from bursting. So now let's consider this question. Back to our IV bag case study. If you're hooked up to this IV bag at the hospital, in general, the contents of the bag in relation to the blood of the patient should be isotonic, hypertonic, or hypotonic. So if you don't want to see the answer, pause the lecture. The answer is isotonic. Remember, that's when your red blood cells are happiest. In fact, in that IV bag is usually, in general, about a 0.9% saline solution. And that is approximately the same solute concentration that our blood has. Now, plants are different. Plants aren't happiest in an isotonic solution because they have those cell walls. So first off, recall that plant cells are surrounded by a cell wall. And also think about when plants are happiest, when you water them, right? You water your plants to keep their leaves from drooping and wilting. When plants wilt or droop, if you forget to water them, for example, the reason they look bad is because there's a drop in the turgor pressure in their cells. We water plants with pure water, right? And that pure water is going to enter into those plant cells because pure water is a hypotonic solution relative to the contents of a plant cell. So for plants, they are happiest when we place those cells into a hypotonic solution. We then refer to those plant cells as turgid, which is their normal state. Plants start to wilt when they have an isotonic solution around their cells. And then we refer to them as flaccid. And if we place plant cells into a hypertonic solution, then they're really not happy. All of the water leaves the cell. All of the water leaves that central vacuole that we find in our plant cell. And then we refer to the cell as being plasmolyzed. And you can see these words on your screen: turgid, flaccid, and plasmolyzed. Our next way that substances can move across membranes is called active transport. And active transport allows us to move against the concentration gradient. Active transport allows us to pump our solutes from low concentration to high concentration. But because it's able to do so, it requires energy. And that energy usually comes in the form of ATP. Here's a question. Why does the sodium-potassium pump on the previous slide require energy? Is it because it uses a membrane protein? The next choice is it transports ions. The next choice is it moves solutes against their concentration gradients. And the final choice is all of the choices are correct. Pause your lecture if you don't want to see the answer. The only correct answer is it moves solutes against their concentration gradients. Active transport, therefore, is the movement of molecules from low to high concentration. It requires extra energy, which is typically in the form of ATP, and as such, it requires protein pumps because the protein needs to interact with that ATP in order to pump those molecules from low to high concentration. To go back to our cyclist example, you've got to pedal like crazy to get uphill, right? It takes lots of energy to pedal up that hill. So the up gradient is not an easy thing to achieve. That's why we need to use energy. In review, we've got two types of passive transport. The down gradient is the movement from high to low, and we can do simple diffusion straight through our membrane, or facilitated diffusion, which is straight through a protein. Active transport is the up gradient. It allows us to move from low to high concentration, but because we're going against the concentration gradient, we must use ATP, which is a form of energy that we can use to get work done in our cells. So what would happen if this IV bag had pure water? Would the cells shrivel? Would the cells expand? Or would there be no net change in our cell? Pause the lecture if you don't want to see the answer. The correct answer is the cells would expand if we had pure water hooked up directly into our veins. The next way that we can move molecules across membranes is called exocytosis. This is how cells release large molecules from the inside of a cell to the outside. What about taking in large molecules? This is called endocytosis. Remember, endo means within, so we're taking particles into our cell. This is how some cells engulf particles, which we can refer to as cellular eating. Of course, we put that in quotation marks because it's not really eating. There's no mouth involved, correct? Here's an example for how endocytosis functions in our immune system. As you might know, we have white blood cells, which play a huge role in helping our body stay healthy. For example, here's a picture of a white blood cell that's engulfing some harmful bacteria in our body. Well, remember our friend the lysosome? So these lysosomes in cells are those little sacs composed of membranes that have digestive enzymes inside. They can fuse with a waste product and digest that waste product. Here we can see how that works. That white blood cell is able to engulf those bacterial pathogens, and then it can use these lysosomes in our body and inside that white blood cell to fuse with the bacteria, digest it, and remove it from that cell. A final example of how substances might move across membranes is in the example of a knee injury. Let's say that you hurt your knee and it swells up. You might have heard that it helps to soak in a hot bath of Epsom salt to reduce swelling. What is Epsom salt? Well, it's actually not salt at all, but it's a type of magnesium sulfate, which has a very strong affinity to water. It pulls water towards it. If we soak our knee in a water and Epsom salt bath, then it can reduce the swelling. Why does soaking it in Epsom salt reduce the swelling? Is it because the salt bath is hypertonic to the swelling in the knee? The salt bath is hypotonic to the swelling in the knee? Or the salt bath is isotonic to the swelling in the knee? So pause the lecture if you don't want to see the answer. And the answer is the salt bath is hypertonic to the swelling in the knee. Therefore, water is going to move out of the cell and into that hypertonic environment around the cell. Chemotherapy drugs are those drugs that we give to patients with cancer. They can help fight those cancerous cells from dividing out of control by killing those cancerous cells. Chemotherapy drugs diffuse in to kill cells, but cancer cells are able to fight back. Cancer cells can actually use active transport to try to pump those molecules against the concentration gradient. Therefore, doctors need to prescribe just the right balance of chemotherapy, not too much to make you really sick, but also not, if it's not enough, the cancer cells will just pump the drugs out. In review, here's a nice chart that shows us all types of transport across the cell membrane. And here is a concept map directly from your textbook that shows all forms of membrane transport. Make sure you understand all of these for the exam. A final question from part 1 of this lecture. This solute is blank and its transport blank require ATP. So here's all of your choices. Please take a look at these. It's either polar does not. The next choice is polar does. The next choice is nonpolar does not. And the final choice is nonpolar does. So for this fill in the blank, the best answer is this solute is polar and its transport does not require ATP. Because in the diagram, what we see is solutes are being pumped from high to low concentration through a protein in the membrane. So what we see in this diagram is facilitated diffusion. Consider this question. Energy is only found in living things. Would you agree or disagree? Pause if you don't want to see the answer yet. Disagree is the answer. Energy is found in chemical bonds, and many chemical bonds don't involve living things at all. We find these chemical bonds in living things, but the chemical bond itself is not a living thing. Now we're at the beginning of chapter 4. It shows us a cyclist, and that cyclist is powering their bike through this forest. Many of the spiker's cells are extracting energy from food and using it to power his muscles. But what is energy? Well, energy is simply defined as the ability to do work, or more specifically, to move matter. Here we can see potential energy compared to kinetic energy. Potential energy examples include the chemical energy stored in bonds or concentration gradients across the membrane. Kinetic energy includes examples such as light or sound or the movement of atoms and molecules or muscle contraction. In the diagram, it shows us a cyclist that pedals to the top of a hill. At the top of the hill is where the cyclist has the highest potential energy. But then as they coast down the hill, at the bottom of the hill is where that cyclist has the highest kinetic energy. Energy changes form within any biological system. And in this diagram on your screen, you can see this relationship of photosynthesis to cellular respiration and all of the energy transformations that are occurring between all living things in the system. Notice that heat energy is lost at every step of this process. And this heat energy is disordered energy. Disordered energy cannot be converted back into a useful form of energy. Instead, it just gets lost into the atmosphere. Therefore, we could say that these energy transformations are somewhat inefficient, and yet we need these energy transformations in order to function as living things. Entropy is a measure of the randomness or the disorder of the universe. Since heat energy is constantly being lost to the universe and heat energy is disordered, the entropy of our universe is increasing. And we can see this playing out in this light bulb example, which we're going to have a question about in just a moment. To the right, we see a light bulb falling through space, and we see a light bulb that's just about to explode as it hits the ground. And then finally, we can see the light bulb is completely exploded into a million tiny pieces. Assume that stage 2, which is that moment just before the light bulb explodes, and stage 3 is that moment just after impact. At what stage is potential energy the highest, and at what stage is entropy the highest? So you've got 5 choices. Choice 1 involves 2 of these numbers, and so on and so forth. So look through these on your screen, and the best answer is the final choice. That is to say, choice 1, which is where that light bulb is falling through the air, but it is not moments before impact. That is where the potential energy is the highest. And then where that light bulb is exploded into a million little pieces, that is where entropy is the highest. To help us understand how energy gets converted in living cells, we can use the car as an example. We put gasoline in our car. That's the fuel source. That's the energy source. And then oxygen travels in through the front end of our car, through the grill. That oxygen goes into the engine. And the combustion reaction, which powers the car, which provides kinetic energy of movement for our wheels, is going to have heat energy lost as a byproduct. And we're going to have carbon dioxide and water drip out of our tailpipe, right? All of these waste products are going to be poor in chemical energy. Well, cells work the same way, except cells don't use combustion. Cells use cellular respiration to break down those foods, and cellular respiration requires oxygen. Heat is lost as a byproduct, and just like the car, carbon dioxide and water are created as waste products. Before we continue our exploration of how these energy transformations work in cells, we need to define the term metabolism. Metabolism includes all the chemical reactions in cells, including those that build new molecules and those that break down existing molecules. Chemical reactions may require energy input or release energy depending upon the type of reaction. An energy requiring reaction is photosynthesis, and that's illustrated in the figure on your screen. It shows us that energy is going to be required to go in order to build those sugars in photosynthesis. Cellular respiration is an energy releasing reaction because we're going to take those sugars and we're going to break them apart, and we're going to, in the process, create energy in the form of ATP that can be used to do cell work. Most energy transformations in organisms occur in oxidation reduction reactions. On the screen now, we can see a phospholipid bilayer with a bunch of big proteins embedded in it. Oxidation is the loss of electrons from an atom or a molecule. Oxidation reactions release energy. Reduction is the gain of electrons, and it's also the gain of whatever energy was contained within those electrons. Reduction reactions require energy. An electron transport chain which we can see that word on the screen here, is simply a series of proteins embedded in a membrane that participate in sequential oxidation reduction reactions. We're going to see these electron transport chains in both photosynthesis and in cellular respiration, playing a key role in helping these cells sustain life by completing these chemical reactions in useful and meaningful ways. All right, here's our next question. In the reaction at the end of the electron transport chain, molecule X is being blank and molecule Y is being blank. And we simply are looking at this diagram that shows proteins, and at the end of this protein chain, we can see molecule X is the protein and molecule Y is an electron acceptor. It's the molecule being reduced. So if we fill in the blank, the answer is molecule X is being oxidized and molecule Y is being reduced. ATP is the cellular energy currency in a cell, and ATP is actually an abbreviation for a much larger word, and that word is adenosine triphosphate, and we can see that word on our screen. It's a nucleotide that temporarily stores energy. All cells rely on the potential energy stored in ATP to power chemical reactions. Here we can see this 1, 2, 3 phosphates in this tail is why it's called a triphosphate. And this chemical bond has a lot of high potential energy stored inside of it. Removing the endmost phosphate group by hydrolysis releases the potential energy stored in that ATP. Therefore, the cell can use this energy to do work. ATP, however, must then be reformed. Mitochondria release energy from food, producing this ATP from ADP. And notice this one is A, letter D, letter P. So that's diphosphate. Di means two. ATP synthesis is coupled with energy releasing reactions, and ATP hydrolysis is coupled with energy requiring reactions. As an example, we can see here that ATP is energizing our target molecule and making it much more likely to bond with other molecules. We can see a glucose, which is a type of monomer, is being hooked up in a long chain with other glucose. And we know the fancy word for that is a polysaccharide. It requires ATP to build this large molecule out of smaller subunits. Another example, ATP donates a phosphate group to a protein, allowing ions to move across the membrane. This is going to allow us to pump solutes against a concentration gradient. In review, we should understand all parts of this ATP cycle. You should know the full names of ATP and ADP. You should know what the energy is for and where the energy comes from. Here's a question to make sure we understand ATP. Which reaction is likely coupled with the hydrolysis of ATP? Would it be an energy requiring reaction or an energy releasing reaction? And the best answer is an energy requiring reaction. When you're studying for the exam, make certain you can describe the relationship between energy requiring reactions, ATP hydrolysis, and cellular respiration. Chemical reactions in cells must occur very quickly in order to sustain life as we know it. Enzymes are proteins that speed up these chemical reactions without being consumed. These enzymes are required for life. Substrate molecules bind to the enzyme's active site, where the chemical reaction actually occurs. Once the chemical reaction occurs, product molecules are then released. The enzyme retains its original form, and the enzyme can do work again. Note that not all enzymes break a single substrate into two products. This example we're showing you here does break it into two products, but other enzymes combine substrates into one product, for example. To help us understand how this works, I like to think of this as a lock and key. Of course, does every single key fit into every single lock? No. It's all about the shape. Shape matters. One specific key is cut to fit one specific lock. And that's just how enzymes work. Active sites have specific shapes that fit specific substrates. If the shape is correct, the reaction will occur. If the shape is incorrect, no reaction will occur. Enzymes are important because they speed up these chemical reactions by simply lowering the activation energy, which is the energy required to start the chemical reaction in the first place. If we look at the graph on our screen, we can see that in the chemical reaction that has an enzyme, it doesn't take as much energy to get the chemical reaction to move forward. But look at right next to it. In this example, there's no enzyme present, and so it takes a ton of more energy in order to get that chemical reaction to move forward. If all of our chemical reactions had to move forward without enzymes, we wouldn't be able to get anything done. We wouldn't even be able to get off the couch because it would require way too much energy. Only some chemical reactions occur in a cell at a given time. So we could ask ourselves, how does the cell control the rate of these chemical reactions? One way cells control the rate of chemical reactions is by limiting the activity of these enzymes. The first example is called non-competitive inhibition. This is our shape changer. And that inhibitor changes its shape. Therefore, the enzyme can no longer bind to the reactant that it was intended for. The other example is competitive inhibition. This is what we think of as the imposter. In this one, the inhibitor binds directly to the active site itself, so that the intended substrate, the substrate that's supposed to bind there, can no longer bind because the inhibitor is blocking it. Many factors affect enzyme activity in our cells, including pH, salt concentration, and temperature. Changes in pH, salt concentration, and temperature can denature an enzyme. And this word denature simply means the enzyme loses its shape, it unravels. Remember, we learned in our previous lecture, if proteins lose their shape, then they don't work. And the same is true for enzymes because enzymes are proteins. Our textbook gives two examples. On this graph, we can see that depending on the organism, they have special types of enzymes that work best at specific temperatures. For humans, our enzymes work best at our body temp. But for this bacteria that lives in a really hot spring, these hot springs would denature the enzymes for a human or any other organism, but this bacteria has evolved to have enzymes that can function properly at extremely high temperatures. All right, so if we look at this graph, at what temperature would we predict the bacterial enzyme becomes denatured? The best answer is 78 degrees Fahrenheit, because that's where the peak of the graph is seen. Just before 78 degrees is not the highest rate of reaction. Enzymes are everywhere. In one example, we can talk about aspirin. Aspirin relieves pain by binding to an enzyme that cells use to produce pain-related signals. In lactose intolerance, intestinal cells do not secrete an enzyme called lactase, and therefore the individual cannot digest milk sugar. If you've ever made Jell-O with fruit in it, you can't put raw pineapple in Jell-O because raw pineapple contains an enzyme that breaks down protein. Now, if you put canned pineapple in Jell-O, that gelatin will solidify because canned pineapple has been boiled for canning to kill bacteria, and that high temperature denatured those enzymes. Our final question, what is the role of an enzyme in a cell? Is it to become consumed during a chemical reaction? Or is it to speed up chemical reactions? Or is it to increase the energy required to make a reaction occur? Or finally, is it to provide energy to the cell? The best answer is to speed up chemical reactions. The primary role of enzymes in every cell is to speed up chemical reactions so that living things can function efficiently. In summary, enzymes are molecules that speed up biochemical reactions. Enzymes lower the activation energy that's needed to start a reaction. Enzymes are shape-specific. Enzymes are not consumed or altered during a biochemical reaction. And finally, enzymes are affected by pH, salt concentrations, and temperature within a cell. If enzymes are denatured, they no longer will function. All right, that's it for Chapter 4. We've now discussed membranes and how molecules move across membranes. We've talked about ATP, which is the form of energy that cells use to get work done, and we've talked all about the importance of enzymes. That now prepares us to talk in detail about metabolism in living things.